Hello, my name is Stefan Braam, founder and CTO of Cellistic. Today we'll talk about our off-the-shelf allogeneic platforms to accelerate allogeneic cell therapy. So we really started with the observation that cell therapy has been great curing patients that previously had no real chance. And so what we are seeing early 24 is that things are progressing the way we were hoping. So I think it's very interesting and great that Carvecti is now approved second line, so more and more patients get eligible. We also, for the first time, really see some really interesting proof points in solid tumors. So there's a study from Penn University where glioblastoma was essentially melting almost overnight following CAR-T treatment, so real great proof point. We also see, unfortunately, some safety concerns popping up, most likely due to the fact that transgenes are integrating randomly in the genome, forcing us to think about genetic engineering and come to better products that are more safe. So we have a couple of industry challenges, which some are good, some are bad. So a growing number of indications, so we need manufacturing capacity up. We see clinical data that justifies earlier lines of treatment, so we need to get manufacturing capacity up. Quality concerns, so we need to build better products. The manufacturing costs are obviously high because of the autologous way that we do things, and we see reimbursement challenges as a result. So I think we have to be honest that autologous cell therapy is actually a little bit more like a process than a product. So the way to solve this, the way we think about it is, how do we build a product? So in our opinion, there are two ways of doing it. Both rely on allogeneic. One cell source is donor-derived allogeneic. The other cell source is iPSC-derived allogeneic. And I'll tell you why we think that iPSC is the route forward. So first of all, when you go donor-derived, you will always have multiple donors. So you will have comparability challenges. With iPSC, you pick one patient, one donor, one product for the lifetime of the patient. With respect to the quality, the gene edits are now done differently. So we are no longer doing this at scale in every batch. We do this actually once in a single cell, precisely characterize that single cell, and we actually have the flexibility to do now much more sophisticated things. So we can put them in a specific locus, we can engineer where we want due to CRISPR technologies. And expansion is no longer a real problem because exhaustion, like we see in adult cells, is not a problem because iPSCs expand very well. During a differentiation process, you expand very well. So you don't have to exhaust the terminally differentiated cell anymore. So big advantages on iPSC. I also have to acknowledge that iPSC is quite tricky. So I've been working on that 20 years, so got some scar tissue on my body. So what we have done with Cellistic, and this is where we are really different, so we built iPSC manufacturing platforms. And we simply do that. We work with therapeutic developers that then convert from a platform to a product. And when I talk about platforms, I really mean platforms. So we start with donor material, we edit that, we reprogram that to get to a product-specific master cell bank. And from that product-specific master cell bank, we manufacture the target cell at scale in a reproducible way. So focused on immune cells, so NK cells, alpha beta T cells, gamma delta T cells, macrophages, and I will give an update on our Treg collaboration with Quell Therapeutics. So a manufacturing platform now looks like this. What you will see here is actually very similar to a biologics manufacturing process. So on the left-hand side, cell line development, like you do in a biologic. So you build the cell line, which becomes the basis of the product. You take that into manufacturing, you produce batches of cells. So very different than a classical cell therapy manufacturing process. So we have two platforms, as I mentioned. So Pulse is our cell line development platform. So what we have now done is to really take state-of-the-art technologies and to think about how to do this in the right way. So we picked the right reprogramming technology, we picked the right gene editing technology, we picked the right gene cell banking technology. We put in place a lot of automation to do this in the shortest time frame possible because you don't want to culture cells for 1.5 years because that will do weird things to cells. Nobody wants that. So we shrink that timeline with automation, very advanced workflows, full quality oversight, GMP principles throughout, analytical testing, et cetera, et cetera. So what is quite exciting, what we announced last week, is that we are now introducing our proprietary CRISPR technology in this platform. So it's our technology, the materials are sourced by us, so we enable our customers to immediately jump into this technology and don't have to shop around in this quite complex field. From a technical perspective, what is really interesting, if we benchmark this technology to competing technologies, and this is public data from companies, then we see that we are getting knockout efficiencies that are a little bit better, which is nice, but where the real difference is with knock-in. So we are seeing knock-in efficiencies up to 50%, where competing technologies are struggling to get to 10, 20%. And this is actually quite important, because if we think about an allogeneic candidate, then we typically see in customer conversations HLA knockout, maybe class 1, class 2, maybe something to avoid that cell immediately get eaten by an NK cell, maybe a CAR or a TCR and another potency announcement. And with that, I'm already at 5. There are people that try to push this further to get to a heavily engineered cell. So we are really making now a different candidate here. So a lot of this technology, or all of this technology, has been developed by Artisan Bio. The technology now is in Cellistic. And so when we go further, safety, as mentioned, is important. When it comes to CRISPR technology, off-target is important. So looking at a bioinformatic analysis between Cas9, the gold standard in the field, and star CRISPR, then we see that star CRISPR versus Cas9, Cas9 generates 8 to 23 times more off-target effects, so undesired effects somewhere in the genome. If we map those effects to where they are, because there is a difference between a gene desert and an oncogenic gene, then the chance that we are hitting high-risk regions is approximately 10-fold lower with star CRISPR. And the reason for that is that Cas9 actually has a PEM motif of only 3 nucleotides. Star CRISPR is a next-generation nuclease, has a PEM motif of 4 nucleotides, so it's simply more precise. And that is very important, because that means that you have to screen less colonies to find the right one. So after building that cell line, that gene-edited cell line, we bring that into a manufacturing process. As mentioned, we are working on NK cells, on alpha beta cells, gamma delta, and macrophages. We have all the analytics in-house to measure identity, potency, functionality. And I think what is really important here, because this is a new modality, and to be successful in a new modality, in this case you want to bring cell biologists together with process engineers, together with manufacturing people, all in the same campus. So that's what we did. We also built our own GMP facility with flows dedicated to iPSC, and we actually believe that this is probably the first GMP facility completely dedicated to iPSC technology. So when we think about such a process, there is no surprise on the top. So you want to have it scalable. You want to have it robust. It has to be consistent, as simple as possible, cost-effective, well-defined, and regulatory friendly. And so what is really important here, because this is quite complex biology, that you start to think about this from day 0. A classical paradigm of a little bit of research and then scale out, scale up will cost you years, and that's not great, because we want to get these assets into the clinic. And so what we do is actually put teams together of cell biologists and process engineers, where the cell biologists really focus on the simple quote-unquote task of understand human biology, understand what signal is needed to make mesoderm, to make hemogenic mesoderm, to make the right cell type, and where the process engineers are thinking about, okay, what have I learned in biologics, what have I learned in gene therapy, what have I learned in cell therapy, build that process and to bring that into GMP. So as mentioned, we have our own GMP facility. We are working with typically well-defined, acceptable or accepted manufacturing technologies from vendors that you will recognize. The GMP facility is, as mentioned, optimized for these type of processes. So we have grade AB for small-scale open operations. We have grade C for bioreactor operations, dedicated fill and finish, dedicated labeling and inspection, cryopreservation, so to run these processes as effective as possible. And to give you an example about such a manufacturing process here, high level, the NK process. So what we do, take iPSC out of liquid nitrogen, we expand them, we inoculate them in a bioreactor, in a stir tank bioreactor. We let them differentiate to mesoderm, to homogenic mesoderm, to HSC, and we harvest those HSC. And in that step of the process, we get a nice 10-fold expansion. Then we take those cells out again, and we let them go to lymphoid progenitors and to NK, and then we get a 30,000-fold expansion. So basically in that process, what you now see is that you get a 30,000-fold expansion from iPSC to NK. So while you scale up, you get to larger volumes. So the way we designed this process is to get ready to a phase 1, typical batch size of 75 billion, so that you can actually treat quite a number of patients. We designed this in such a way that it's completely free-to-free, that is not unscalable at any part of the process, because a 75 billion batch size is a starting point, not a commercial batch yet. We use chemically defined media. We make sure that it works with different cell lines so that a therapeutic developer can actually plug into this platform and build with confidence. So when we take those cells and characterize them, we obviously make sure that they do what you would expect from an NK cell. So when we mix them with tumor cells like K562, then we see within hours that they kill, as we hope. We see that they secrete the right type of cytokines. We do extensive flow panel, which I cannot show today, but happily to talk about it after. So we really make sure that we have the right starting point for a therapeutic developer to now build a product based on this. Take these cells and now put in a car or any other type of creativity that you might have. We can actually build a therapeutic together really, really quickly. And so then that brings me to Quell. So this was announced 2 years ago. Great project. Our regulatory T has been probably the biggest challenge that I've faced. Because what we're now doing, starting with that iPSC, we are going to that same HSC, but now we go to a pro-T cell, then to a CD4 ISP, then to a DPT, then to a mature T cell, then to a CD4 cell. So this is actually a cell that in embryonic development actually arises quite late, so we need to play a lot on the biology to get there. It's great to work directly with a therapeutic developer because a company like Quell knows very well what a good cell looks like. So what we've done with them, a lot of flow profiling, Treg activation assays, suppression assays, cytokine secretion, and the most stringent test that they could come up with, a specific region in the FOXP3 that should be demethylated when you arrive at that Treg, because the region should be accessible and FOXP3 needs to be expressed. So that's the most stringent test. And what we see in that blue graph, in DPT cell, it's not demethylated. In CD4, it's not demethylated. Suddenly there. So it passes now the most stringent test. So we have now pioneered biology to get towards Treg. And I think this is an interesting proof point where we are very different from a classical CDMO that would never do this type of work. In summary, on the business model, so we have seen fantastic leading therapeutic developers like Stefan, who speaks after me, that basically build everything themselves. So the iPSC engineering, the differentiation process development, maybe the engineering, the manufacturing. And so what we've seen is that they had to have a lot of grit, a lot of resilience, a lot of time, and a lot of money to get there. And it's great that these therapeutic companies are now delivering. What we are now doing with Cellistic is something a little bit different. We built a manufacturing platform, we make that accessible, and as a result, we believe that we can cut that time to an IND from 5 years to 30 months, that we can cut those investments of north of 100 million to 20 to 25 million, basically enabling therapeutic developers a new way to iPSC allogeneic cell therapy. So in summary, oops, sorry for that. I hope I convinced you that iPSC is the most attractive starting material for allogeneic cell therapy. And we enable therapeutic developers with our Cellistic platforms to expedite that product development, to avoid common pitfalls in that iPSC biology journey, to exploit our pre-established platforms that are scalable and that are predictable. And with that, we really bring standardization and manufacturability in the project from day zero. That I will end. If there are any questions, please find me after my talk. Thank you.